Today, there is one cancer, and only one, in which only 10% of patients will survive for 5 years after diagnosis. And in this cancer, even those patients who are diagnosed with what we call localized disease, that is disease that is confined to its site of origin, that even only 40% of those patients who undergo surgical removal of their cancer and get chemotherapy will survive for 5 years. And that cancer is pancreatic cancer. So what these grim statistics tell us is that even in those cases where we think the disease is still in the pancreas, it's spread elsewhere in the body. And perhaps more importantly, that disease is resistant to the chemotherapies that we currently use. And so we need new effective therapies. Work in many labs over the last 30 years or so, which has really accelerated maybe in the last 15 years, has demonstrated that targeted therapies that go after specific factors that are required for the formation, growth, progression, and ability of cancers to kill the patient, that going after those targets can be effective. And in the Lewis lab in the Department of Molecular Cell and Cancer Biology here at UMass, we spent the last several years trying to identify those factors in pancreatic cancer. So we've identified some of these, and we've spent the last few years focusing on one particular factor, a protein called mTOR. So we showed using genetic tricks that if we get rid of mTOR in an animal model for pancreatic cancer, that we can block the ability of cancers to form. And importantly, if we take a model where we combine multiple mutations, that gives us aggressive disease, that even in that context, if we block mTOR activity, we can significantly extend survival. So it's important to note that even though we extend survival in these settings, that the mice still die from pancreatic cancer. And so this brings us to the idea of combination therapies. That is, can we identify other drugs that if we give them at the same time as a drug that blocks mTOR activity, that we can get around the ability of pancreatic cancer cells to escape their need for mTOR. So I like to think of this in a way as similar to the way we used to treat HIV patients. When we first developed drugs to treat HIV, we went after one factor, and we could see viral loads go down, and then they would come back up, and we'd see either onset or progression of AIDS. But now we treat patients with multiple drugs that target multiple parts of the virus at the same time. And in so doing, we block the ability of the virus to develop resistance or to escape the effects of the drugs. And so we wanted to do something similar for pancreatic cancer. So we've performed a screen to identify drugs that will cooperate with an inhibitor for mTOR. We've identified many such drugs, and we validated some of them in a collection of pancreatic cancer cell lines and other experimental models. And so what we're interested in understanding really are three things. One, can we identify these combinations? And the obvious answer to that is yes. Can we identify markers that predict which pancreatic tumors will be sensitive or be killed by these drug combinations, and which ones will not be killed by these combinations? And can we use this information to begin to understand why some tumors will be killed and why others won't? And we can use this information then to tailor therapy specifically to individual patients. So we have made some progress in this. We have identified pancreatic cancer cell lines and pancreatic tumor explants that are sensitive or get killed by some combinations, but not by others. And so we're confident that we can identify those markers and begin to understand how or why these therapies work in the settings that they do. So this is going to be a very significant undertaking, but we are very excited by the possibility to develop new novel therapies for pancreatic cancer, a disease for which this is very much needed.